So in the 1940s, Barbara McClintock was studying maize, corn, and she was trying to map a gene, meaning find out which chromosome this gene was located on. And she would set up some experiments and do some crosses, and she would have some data, and she would say, okay, it seems to be the gene is definitely on chromosome 2. And then she would do more crosses to confirm it, and then she would look at the data and she'd say, oh my gosh, I was totally wrong, it's on chromosome 6. And she kept doing it, and she finally realized that she wasn't making a mistake. The good thing was Barbara McClintock was very good as a scientist, and she finally realized that the gene she was trying to map was moving. Now in the 1940s, this sounded like she was just crazy, but she was very good, and eventually she was able to really demonstrate what she won the Nobel Prize for in 1983, and what we call transposable elements, or transposons, or sometimes they just call them elements, which is actually kind of confusing, but I'm going to call them transposons. Transposons are a little linear piece of DNA, and typically it contains, there are certain genes that typically are on that piece of DNA, and that DNA can actually insert itself onto a chromosome of the host cell. Transposons live inside the cell and they never leave. They don't leave the cell. It's not like a virus. A virus is similar, but it eventually leaves the cell. A transposon stays inside the cell, but it can be inserted into one chromosome and one gene, and then later it can move to another chromosome in the same cell. And so these things get copied during DNA replication, and so they get passed on to daughter cells during cell division. And so cells that have transposons will still have transposons after cell division because they'll be copied when the chromosomes are copied. They're very well known in bacteria. In bacteria, they're called insertion elements, IS. In Drosophila, of course, in maize. In Drosophila genetics, they're very well known, transposons. And you'll remember that ALU is a repeat that occurs all over the human genome. And ALU, we think, it comes from a transposon that no longer appears to move. That meaning that the ALU that we have seems to stay where it is from, as the cells go through generations. So here's what transposons do. On the right side, here's a kind of a very rough drawing of what a transposable element looks like. At the very ends, it has these repeat sequences. And then in this yellow section in the middle, that's where there are certain genes that are usually encoded there. The genes that are on a transposable element are usually genes that encode enzymes that are necessary for the transposon to insert into the chromosomal DNA and for it to excise itself from the DNA. And so they don't show it here, but the enzyme, the genes that encode those enzymes necessary for the transposons to be able to move around. When a transposon moves, we call it a jump. So transposon jumping. They sort of jump into a chromosome and then jump out of it and go into another chromosome. But they stay within the same cell. If they insert into a gene, it can mess up the gene just for that cell, though. Anyway, on the left side, the enzyme that is encoded on the transposon itself will cut the DNA, and then the transposon will insert itself in, and then the DNA polymerase that the cell produces will fill in these little gaps. And then excision of the transposon fundamentally involves another enzyme that just makes two cuts, and the transposon is released. And sometimes it leaves the chromosome kind of broken at that point. Sometimes when the transposon leaves a site, it doesn't necessarily join the pieces together again. The cell has some ability to do that, but sometimes it just leaves broken chromosomes. One interesting story of transposons that relates to real life, not just research kind of things, is grapes. What we call black grapes are those really, really dark purple grapes. Those are like wild type grapes. That's the actual original version of grapes. And they have a gene called VVMYBA1, which is shown here in green. And that produces these dark pigments that are in that black grape. Those are really good for you, those antioxidants, pigments. Then it turns out that green grapes, when they studied the genome of green grapes, they actually found a transposon right next to the gene VVMYBA1, and that disrupts expression of that gene. I haven't looked into it, but maybe it messes up the promoter or the enhancers. It's something, it affects the expression of the gene. And so those anthocyanins are not produced in the green grapes, and so you don't get that dark pigmented color. Then they studied the genome of red grapes. In red grapes, they see evidence that there had been a transposon next to the VVMYBA1 gene, but it has excised and left. So it's somewhere else in those cells, but it's not there anymore. And so there's like a little scar, a little piece of DNA left behind that they can recognize as a piece of the transposon. But because the large transposon is gone, there's less disruption, and so you get partial expression, or you get some, not full expression, but a low level of expression of the gene, so you get some of the pigments, and that's what we call red grapes. So that's kind of interesting, I think. So like I was mentioning before, ALU is this very short repetitive sequence. It's a sign, short interspersed element. And an element, E is for element, so transposable element. And it appears a million times in the genome, but they thought it was an ancient transposon that can no longer move. And they still think that, but so a repetitive element called a line was found inserted into the clotting factor gene of some hemophiliacs. Hemophilia is a disease where either one of two different clotting factor genes is not functional. Usually due to a mutation, like a point mutation, a single base substitution, or something like that. Well, these two patients, and they weren't related, so two different insertions, but both of them had this long sequence, and it's not something that DNA polymerase is going to accidentally make a mistake on one base pair. It's like 1,000 base pairs of a very recognizable sequence. But neither of their parents had that in their gene. What it would suggest is that somewhere in gametogenesis or in the early embryo, a line from somewhere else in the cell jumped into that clotting factor gene. And so that makes us think that maybe these transposons that are in the human genome do move. Maybe not a lot, but maybe they do move sometimes. We at least have to consider that possibility based on this data. All right, what about repair systems? There's a couple different repair systems. One is called photoreactivation. You remember those pyrimidine dimers, those thymine dimers that join together? Two thymines that are next to each other in the DNA when UV hits them. Photoreactivation is a repair process. It's only in bacteria and yeast, and it can actually break that covalent bond between the pyrimidine dimers. So it's a direct repair system. Nucleotide excision repair that would repair pyrimidine dimers in other organisms that are not bacteria or yeast. So for example, humans. If we get pyrimidine dimers because of UV light, when UV light hits your skin, hits your eyes, hits everything, it creates these covalent bonds between thymines. It bends the DNA backbone and it increases the chance for mutation during cell division, during DNA replication preceding cell division. And nucleotide excision repair cuts out one of the strands, not just the dimer, but like 20 or 30 base pairs on one strand. It leaves the other one there. And then it just uses that strand that's left behind as a template, and it just copies it back with polymerase. There is a disease called xeroderma pigmentosum. We usually call it XP for short. And they have a mutant allele, a defect, homozygous recessive typically, a defect in one of the genes that encodes these enzymes. So their repair system for DNA for skin, mostly skin damage, is not working properly. So here you have a picture of a girl. It looks like she just has freckles, but in fact these are all lesions, precancerous lesions and damage caused by sun, which cannot be repaired the way that most of us can repair it. It gives you an idea of what would happen if you didn't have those repair systems. Everybody would look like that. So the sun does cause damage, and most of the time your cells are able to repair it. The other kind of repair is called mismatch repair. It's a specific kind of nucleotide excision repair, meaning there is going to be one strand that's removed and then just rebuilt. But mismatch repair specifically refers to the repair system that happens when polymerase makes an error during replication. So let's say polymerase makes a spontaneous error. It could be because of a tautomerization event. Whatever the reason, if polymerase makes an error, and in this picture here, let's say polymerase made an error, there was a G and a T in the new DNA. All right. Now the original, the template strand is green or gray, and the new DNA is shown in pink. All right. So T was the template, and then polymerase added the wrong nucleotide G. So how will that be repaired? What will happen is the strand that has methylation, and you'll remember methylation is added to DNA. It's very normal. It's part of that epigenetic mark. But the template strand of DNA will have much more methylation than the new strand, because the new strand hasn't been there long enough to get methylated yet. So the enzymes for mismatch repair will remove the undermethylated strand, meaning the newer strand. And so you can see here the methyl group shown in red, this little red dot down here. That indicates that the gray strand is the methylated strand. So the enzymes will remove, it'll cut the DNA in a couple places. It'll remove all that section, and then it will just resynthesize it. And it almost never makes the same mistake again. So now you have the T with the A instead of the T with the G. So that's called mismatch repair. There are, like I mentioned already about xeroderma pigmentosum, there are other disorders related to, some of them related to cancer, and I've circled a couple of these. Genetic diseases caused by defects in DNA repair. And the ones that I want you to recognize, xeroderma pigmentosum you need to know for this chapter. So freckle-like spots on the skin, predisposition to skin cancer. There is another repair system defect that is related to cancer, except it's colon cancer, hereditary non-polyposis colon cancer, HNPCC for short, and then one called Li-Fraumeni syndrome. So we're going to be talking about HNPCC and Li-Fraumeni syndrome when we get to the chapter on cancer, which is chapter 16. But it's not unusual for some types of cancer to be caused by defects in DNA repair. All right, so xeroderma pigmentosum, make sure you know a little bit about that. for this chapter. And then we will see these other two in the Cancer chapter. And you don't have to worry about the rest.